There are a number of ways to get genetic variation. Remember, variation just means a change in the number or combination of genes. One way for genetic variation to occur is gene flow, when genes move from one population to another. For example, if you moved to Latvia, you would take your genes with you, and if you had children there, some of your genes would combine with your Latvian partner's genes. That leads to another way genetic variation can occur. Sexual reproduction causes new combinations of genes to form. Finally, mutations also cause variation. As you learned in the genetics unit, mutations are random changes in DNA or chromosomes. Sometimes when genes mutate, new genes or gene combinations are formed, which leads to more genetic variation. The total number of genes in a population is known as the gene pool. The gene pool can increase or decrease as genetic variation takes place. Genes move in, genes move out, or new gene combinations are formed.
Genetic drift is one of the mechanisms of evolution because it tells us one way in which evolution can happen. I call genetic drift survival of the luckiest. It's when some members of the population do not survive because of chance, a random event, and not because of fitness. In the example shown, the green beetles are killed because they were in the wrong place at the wrong time. The brown beetles were not better adapted, but there were more of them that happened to survive. Future generations, there will be more brown beetles because there are more of them to pass on their traits. It's not a better trait for this environment. Again, they were the lucky ones that were in the right place at the right time to survive. A special type of genetic drift occurs when only very, very few organisms survive or are the luckiest to survive. It's called bottlenecking because the population narrows or becomes smaller like the top or the neck of a bottle. This can significantly change the gene frequency, especially if some traits are not present at all in the small group of survivors. When the population builds back up, they will look like the small group of survivors. Again, survivors don't survive because they are better adapted to the environment. This is a type of genetic drift, so they are just the luckiest to survive. Oftentimes this happens because of a large environmental disaster, maybe a fire or a big storm that kills off many of the population.
The founder effect is also a type of genetic drift. When you think of finding something, you go and look for it. So in the founder effect, members of an original population go to find a new place to live. So a few members break off to find a new colony. As a result, you end up with a smaller population size and reduced genetic variation from the original population. So with founder effect, you might start to see some weird things that get passed on from the original generation. This is because the population is not large enough to weed out those weird traits. There's not enough genetic variation or genes to help weed them out. So this is an example of polydactyly where you have extra fingers and extra toes.
Okay, so there's two types of speciation, or way new organisms form, that you need to know about. The first is gradualism. This is when there are really small changes over a long period of time, eventually leading to a new species. The next type is called punctuated equilibrium. This is when there are big bursts of change and then long periods of no change. Pause the video and draw the two types of speciation at the bottom of your sheet for question 6. Examples of these two types of speciation can be seen here in the butterflies. On the left, the butterflies are going through small changes until finally 2 new species are formed at the top. On the right, however, they make drastic changes all at once with long periods of no change, and then eventually the new species are formed at the top. The next topic we need to discuss is called adaptive radiation, First, let's take a moment to kind of dissect that term. So adaptive means to change. Radiation means to move out from the center. So this is when you have a single ancestral species spreading out into different environments, and they adapt into new species. The example that we like to bring up is like the finches from the Galapagos Islands. They all come from a common ancestor, but as they flew out to different islands, which had different environments, they adapted to their new environment in order to survive. I like to think of this kind of like a radial tire, also radiation. The spokes start in the middle at the hub. This is like the common ancestor, and then they move out to the edge. This would be like all the new species forming. The next pattern of evolution is called divergent evolution. This is when species that were once similar to an ancestral species become different. Let's look at the word diverge. This means to separate or move away from something. So the example here is the forearm bones of these 4 organisms. They all have the same bones from a common ancestor, but have become different over time, creating homologous structures, which we've discussed before. The opposite of divergent evolution is convergent evolution. When something converges, it means to come together. This is when totally unrelated organisms evolve to have similar traits. Examples of this are the giant armadillo, the giant pangolin, the giant anteater, and the spiny anteater. Even though they look similar, they actually don't come from a common ancestor. They just evolved this way to adapt to an environment that's similar, even if it's across the globe.
Patterns of evolution can also be observed through natural selection. Here is an image of ranges of phenotypes in bell curves. The image at the top shows you a range of light to dark fur color. And on the bottom are 3 images that show you different variations on natural selection. The first one, directional selection, is like when you turn on your directional signal. You either move to the left or to the right. In this case, the whole curve is shifting to the right, and it's favoring the darker phenotypes, the darker fur color. This curve could also shift to the left. Then there's disruptive selection. Imagine your younger relatives decide they're going to come in and jump on your bed. This is extremely disruptive. So you're jumping on the middle of the curve and getting rid of the middle phenotypes. So you have just the light and the darker phenotypes, the extreme phenotypes. Stabilizing selection is when you go to stabilize something. So you're putting pressure to squeeze the curve to the middle. So you don't end up with extreme phenotypes. You end up with intermediate fur color, like a medium fur color phenotype. So we're going to focus on directional selection. On your note sheet, it asks you to write down an example of directional selection. So you should look at the phenotypes of the mice and their fur color and observe how they change. So directional selection occurs when selection favors one extreme phenotype over the other. So in this case, shift from light-colored to dark-colored fur color. Another type of natural selection, stabilizing selection, occurs when selection favors the intermediate or the middle phenotype over the extremes. So we don't have a light or a dark colored fur color anymore, just an intermediate or medium fur color. There's pressure or environmental pressure being put on this mouse population, so only the intermediate phenotypes are selected for. So another example, disruptive selection, occurs when selection favors extreme phenotypes over the intermediate phenotypes. So you smush out the middle phenotype and you're looking at only light color and dark color phenotype fur colors.
If 2 groups of the same species are not able to mate together for a long enough period of time, each group may evolve different traits and therefore different genes. At some point, they may become so different that they'll be considered different species. The way scientists decide if they are different species is an easy test. If they can mate and produce a fertile offspring, then they are considered the same species. If their genes on their DNA have changed so much that they can no longer have an offspring, then they are considered different species, and speciation has occurred. So we now know that speciation can happen when two groups of the same species can no longer mate together. Geographical isolation is one way that organisms can be prevented from mating together. Geographical isolation is when two groups of species are separated by geography. The groups may be separated by a canyon, a river, or a mountain. Can you think of any other ways the groups may be geographically isolated? Once the groups are separated, they can only mate within their smaller group, and any adaptations that occur will be from changes in the organism's DNA. As both groups' DNA changes, they eventually become different species.
So now you know that one of the consequences of geographic isolation is the formation of a new species. This has a special name, allopatric speciation. When populations are separated geographically by mountains or rivers or canyons, genes can't flow between populations, and eventually the organisms are so different that they can't mate anymore and they become a new species. Another way new species sometimes form is through reproductive isolation. This happens when organisms become so different that mating doesn't or can't occur anymore. Organisms can develop different mating rituals or have different mating times, or the sex organs have changed so much that they no longer fit. New species form that way.